this one. Here we go. We're already laughing. Look at that. It's just right off the bat. So yeah, welcome. Good to see you. We're racing each other for who can have the most grey hair. My goodness, I'm still slightly older than you, at least. So
yeah, you're older, but I guess I've known you for 24, 23 years now.
Yeah. Did you start right out the gate? You were one of Mark's first, weren't you?
At Berkeley. So I showed up to Berkeley in 2000, but I joined the lab in 2001. So
Well, yeah, right. So you were exactly. So you were right out the gate. Yeah, because I was thinking about that recently because I was listening to Charan's podcast with what's his name, Lex Friedman. Good chat actually, and so like reliving all of those days, and it's just like, oh yeah, he was I guess he was just finishing up his. No, he finished his PhD, and he must have been officially a postdoc. He was like number 2 behind Clay, I think. So it was Eric, then no 3, Eric, then Clay, then Charan, then Bart. I think it was. I never
Bart
quite.
was first because Bart came from Penn.
No, but they were all Mark's postdocs.
Yeah.
When they all came at the same, for me, they all arrived at the same time, but they had a slight order in Mark's life, right? But they all had been Mark's postdocs at Penn before they joined Berkeley. But it's, yeah, so it's funny. And yeah, that was 23 and a half years ago. Well, there we go. So, Sonia, how you been? Are you still doing mostly fMRI? Are you doing?
Yeah, I'm doing mostly fMRI. I'm a staff scientist in Peter Mahoney's group, and in that role, I get some time for my own research and a lot of time just making sure everyone else here, supporting the research and training of everyone else. So, as I'm talking, I'll be some of it my own stuff, some of it just like, hey, I did this project with so and so, and this project with so and so.
Good. Yeah, good point. So if you have anything, we make sure that you can. I think you should be able to share screen. Just give it a go to make sure it's ready to go.
Yeah, I think I have the button. So
Yeah, perfect.
No, sharing is not turned on, so you'll have to.
Yeah, it's not on.
Yeah, it says host isn't allowing multiple sharing for this.
Oh my goodness. Hang on.
Yeah.
Why isn't it just an obvious button? Let's share. One participant can share. Sorry, people listening. All right. Well, while you're describing something, I'll try and figure out where the button's gone. Host tools. It must be under host tools. Allow all participants to share screen. There it is.
Yay.
Why wouldn't they just put that under the share button? Who designs this stuff? Amazing. And we worry about artificial intelligence taking over the world. Not if it's designed by us stupid monkeys, it won't. It's much more likely to make smoking holes in the ground. Right. Anyway, so cool. Well, let's go. I mean, do you want to go through your list first, or do you want to just dig straight into the stuff that I was suggesting about?
Maybe you start with one of your questions, and we'll see.
Yeah. So questions as much as the things that I think it could be useful for us to come up with a list of things that we think are nice to have versus essential to have for before, during, and after scan measurements. Obviously, in scanning is the most complicated one. So you've heart rate, pulse oximetry, and chest motion are so easy. It's silly not to, whether or not you need them, right? So I think mostly these days, I think the data-driven methods are actually pretty good, probably better than any sort of model based on like chest motion, like RVT, things like that. So I think things like Rapid Tide do a pretty good job of pulling those things out. But in a pinch, why not have those data in case you need to go back to them?
I mean, I feel like there's always like, what to collect and how much effort it is to collect. And if you have an MRI center and the MRI center, is used to fMRI, there should be a pulse oximeter and a respiratory belt. And it is just so easy to collect that data that, compared to the cost of the scan, the cost of setting that up and keeping it running is just so cheap that there's no reason not to collect pulse and respiration. And I'll say also, like, it's not just about noise removal, it's about behavioral monitoring. I mean, I don't even know if you remember this. Like one of one of my PhD projects was I was doing HRF variation, and there was some of the older studies on nonlinearity of the HRF, and I was going to see it with aging. And I was doing like 1 second, 3 seconds, 6 second, flashing checkerboards. And it turned out that some people just hold their breath for the length of the checkerboard. Um,
Oh,
and
right. Yeah, 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 yeah. I do
and
remember.
there's no noise removal method that will ever fix that.
Yeah.
And if we hadn't collected the data, I would have shown a great effect because the signal gets bigger as you see the checkerboard longer, but it was a breath hold. And
And right. So you have essentially two tasks going on. And even if you'd had a data-driven method, let's say you didn't have chest motion,
yeah,
would there be, maybe with ICA or anything, could you have pulled that out and worked out that they could have been holding their breath?
maybe we could work out they could hold their breath, but it would be more subtle.
Yeah. And you
Um,
would never know for sure that they were holding their breath.
yeah.
Wow.
Like
Okay.
you would see a global effect. I mean, the big thing is you would probably see a global response. So if I looked beyond visual cortex, I would have noticed, hey, the whole brain is changing.
Okay, well that's super important because even if you're doing something like, I can imagine, obviously they weren't told to do this. So what is the cognitive load that when somebody's attending to a task, when they're very, very well on task, ironically, they will hold their breath because presumably that's the same thing that can happen when somebody's doing a complicated solving a complicated problem or watching a movie where there's some suspense, right? Naturalistic viewing. So, okay, so then you've just made the case for me. Then we have to do chest motion, chest monitoring.
Yeah. And it's just so easy. I mean, like
Yeah.
I mean, I know you've talked about the harder things like CO2 monitoring, but like chest and pulse are just so easy to collect
Yeah.
that there's really no reason not to.
Right. So treat it less it'll have physiologic effect, but treat it more, as you say, as more as a monitoring device for what is the person doing. Treat it more like a camera uh, for monitoring what people are doing. Okay, that's super cool. I hadn't really fully thought that through, but that's a really good point. Like that one, like that. And I guess again, the pulse ox gives you an additional degree of freedom there because
Yeah.
obviously the pulse and the heart rate variability will vary as somebody's changing their behavior. So it's super important to
Yeah.
know for those periods what's going on. So if you have a primary hypothesis that somebody might hold their breath during a heavy cognitive load or when they're really on task, then you also want to know what's going on with the heart rate and the variability at the same time. Dynamite. Okay. What about attention then?
Um, so I mean, this is sort of one of those things that it depends partially on your task. So like if you're doing a task fMRI, attention might actually be explicitly measured in their responses. Um, but I mean, it's one of those things like I think eye tracking is great, and I think if you can do it and set it up, it's a great thing to add. But it adds time. So
Would
for
you do it as pupillometry, just as basically
yeah,
like sort of
yeah, if you want to do like a pupillometry measure, that's could be a decent proxy of attention.
Mhm.
But setting up an eye tracker and getting accurate pupillometry is going to add some time to your scan. So maybe that works with healthy volunteers. I'm not sure that's ever going to be realistic in like these large inpatient studies. So I love
Unless it
it.
could be set up in a more passive way, like with an
Yeah,
indoor camera, which just happens to already
yeah.
be ready, finding the eye so that you don't have the additional setup.
Maybe something like, and a couple EEG electrodes could be faster, but that's expensive and harder. So
Right, I was going to suggest that. So yeah, because you've now got high impedance electrodes, you've got to have the
yeah,
conductive gel, presumably to get a good
yeah,
signal, etc., etc. And you also have the issue of where you put it, so that it
yeah,
doesn't
yeah.
reintroduce a bias, right?
Yeah. I mean, I think there's definitely studies where that's the right way to go. But if we're talking about like must be part of all studies, I think
Mhm.
that's a bit trickier.
What about something else like muscle tone or galvanic skin conductance, anything like that?
I mean, it's
Sorry.
funny. Galvanic skin response is something that I've like talked about for years, but never actually collected. I think it's a useful measure, but it's kind of like on my list of stuff. It's always like the maybe next study. And maybe I'm wrong there. Maybe I really should be collecting it because again, it's a measure of tone and attention and other arousal measures, and it is relatively easy, but it's just never been something that I focus on.
Right, right. I mean, another issue there is it's electrode, so you have the same issues as with the EG. So now you've got a wire,
Yeah.
you've got a high impedance electrode. Um, I would worry that it would be too slow to be meaningful for the dynamics that we are trying to capture. I don't know, but
Yeah.
might not be my guess. Whereas the pupillometry and the EEG are much more dynamic measures. Well, that's a good question. Okay, well, that's something that I'll have to give more thought to. And this people are watching, there'll be a good challenge to come up with a good, robust, easily set, easy setup for just monitoring attention that doesn't require lots and lots of user interaction. That would be super valuable. Okay. You mentioned gases, expired gases. I kind of go up and down on these. Nice to have if you're doing cerebrovascular, or essential to have if you're doing cerebrovascular reactivity. Do you think that this is necessary or useful for expired gases for?
I mean, I think it's in that same category of like they can be really powerful, but I can't see them like being part of routine studies because they're unpleasant, either nasal cannula or mask.
Yeah.
We're not going to scan, 1,000 people with a mask on.
Yeah,
So
and I just worry that there's not really a huge amount of explanatory power. I used to think that there was, but you're only getting expiration values. The end tidal values might be useful for quantitation for CVR, but I would worry that the complexities of the measurements themselves would be so fraught that you wouldn't get much that's that useful. And I think that there is a case probably where data-driven methods like rapid tide do well enough.
Yeah.
That I don't know that I need O2 or CO2 unless there's some pathology or some reason I need to know this person's expired CO2.
I mean, I think what I also say is, and this might be jumping ahead a bit, but like some of these measures are really good for characterizing the system. So what does it look like when CO2 changes in a way that breathing doesn't? Like what, how does this signal respond? I mean, rapid tide is good, but it's not, I don't think they would say either that, it's a solved issue. So like a lot of these methods and the methods work characterizes the system better. So if we can say
Well,
this,
yes and no, because but then if you do, let's say, breath hold,
yeah,
you don't get an entitled CO2 until the end of the breath hold. You get one
yeah,
data point if you're
yeah,
lucky, right? It's not screwed up. So you could argue that the data-driven methods are actually better because they're at least continually sampling. You're pulling something out of the data.
yeah,
It might not be exactly correct,
yeah,
but short of having a a femoral or a line in an IV sampling, how else could you get
yeah.
blood gases?
But I think still like sort of doing these more invasive studies to better characterize the system will make the data-driven methods better. So I
That's
think,
exactly where I'm from. Exactly. So
yeah,
I think 5 to 10 years ago, I would have been pushing hard for expired gases. Now that I've seen enough, I think sort of believable validation in at least
yeah,
things like blood gases, then I would
yeah,
be okay with that. But blood pressure. Let's move to blood pressure then, because I
okay,
still think that that's an unknown. We might be fixing that with things like rapid tide, but we don't know. So we haven't validated it yet because we haven't done enough studies with blood pressure measurements
yeah.
in the scanner.
So we're doing a little bit of that here. One of the postdocs that works with me, Brock Aiken, is working on this. And I think the big thing that sort of we've come up, sort of from the high level picture, blood pressure mostly matches heart rate. Like if you look in the literature, but
Oh,
not perfectly. So you're gonna get. So one of the questions we're investigating is what happens when blood pressure deviates from heart rate. But sort of, more often than not, they do match. And then the question is like, what are these deviations mark, or how does that, relate to fMRI? And I think it's still
is
an
it
open
roughly
question.
linear over your typical range, let's say 60 to 80 beats a minute?
I don't know offhand. I
Okay.
don't sort of,
So
but
so in other words, we can't disentangle it from everything else anyway, and so there's no point even trying.
yeah, I mean, I think like just sort of like if you correlate the blood pressure signal and heart rate, they're pretty similar, but they're not identical. So then the question is, how much is measurement noise and how much is sort of dissociations? And from the clinical literature, they do separate. So it's not like they're identical,
Mhm.
but sort of your rested heart rate and your resting blood pressure are more similar than that.
Okay.
I
Okay.
think from a practical point of view, there isn't a great MRI safe tool. Like the one we're using, I think it's FinaPress distributed through BioPack. And it's actually quite frustrating because it's this pressure, essentially it's just a pressure gauge, but the ones outside of the MRI have 2 to 3 cuffs, and they kind of do this internal calibration. And the one they sell for MRI just has a single cuff. And there's nothing about it there's nothing in the hardware that says you can't design one with the calibration that's MRI safe.
Right.
They just didn't.
It's silly.
So,
It's a small market, I'm sure.
And they use some magical sort of post-processing to stabilize it, but like, why not just get better data to begin with? So I feel like there's, maybe my opinion on blood pressure will change, but it's right now it's for like general recommendation, it's a hardware issue. There's no hardware that I would say, is MRI safe and you can just buy and use right now.
Well, what about just doing a couple of pulse ox? I mean, I've just tried this recently. I'm waiting to hear back from a couple of people. Just one on the finger, one on the toe, same side of your body. Measure the difference. There's a difference in the arrival time, so then presumably the arrival time will shift with blood pressure.
Yeah.
For the pulse.
I mean, it's absolutely possible. I mean, this relates to kind of where Blaze and Tongue started with their NIRS peripheral measurements.
Right.
And it's funny because they have a system where they're collecting NIRS on like several fingers. I bet the blood pressure data is actually in there somehow.
Probably so.
I don't think they've specifically looked into that, and that might be a cleaner solution with existing hardware. But those,
right.
Yeah.
Okay. Well, there's a student working on that here now. I did send Blaze some data, and I'll remind him, see if he pulled anything out of that. I just needed a peak picking routine to see whether the arrival time shifted at all. I had a couple of silly experiments baked into this test data, and I can do more. Okay, well that's cool. Well, so the jury's out on that one. So you're not currently worried about blood pressure then? It's not necessarily critical. So quick summary. So heart rate, definitely yes. So must have. Chest motion, must have. Attention monitoring, nice to have, leaning towards must have, maybe.
Yeah.
Maybe as we go forward, it'll become must have. I think, as you say, if it was easy to do, we would probably push it into the must have camp. Blood gases seems like nice to have, probably not critical. Blood pressure, probably more like nice to have, also might not be critical. Hopefully we can put that one, hopefully I can work on. What else in the scanner are you interested in trying to measure?
So I think a lot becomes to sort of task design and study, particularly for REST. This is, I think the paper that you originally wrote me about with my colleague Javier Gonzalez Castillo is just monitoring of what people are thinking. Just sort of some feedback either after the scans or even during scans of what's going on in people's minds, because there's so much information there. And it might be, frankly, as big or bigger than some of the clinical effects people are trying to look into.
Are you suggesting that anybody who's doing resting state just have a standard sort of questionnaire and just give it as a default as they come out of the scanner, just to see?
Maybe before, maybe after, or maybe both.
Nobody's doing that right now. There's nothing standard, right? There's no standard pre-post.
There's a few people doing this. So Javier had a paper, sort of, I should look it up, but it's sort of like, what are people doing in the scanner? Ask them. The title is something like that. And it reviews a bit of the literature in that area in general. And one of the things Javier and I have discussed, which I think is actually, which might be very, I'm hoping is actually going to be quite interesting, is like, there's all the early resting connectivity papers. We're talking about like the brains of population A are, differently wired than the brains of population B, but like the wiring is not changing on a macro level. So talking about wiring changes doesn't really make sense. But what could make sense is you put someone in the scanner who, you put someone in the scanner and they think about like, what they did yesterday. They think about, their dinner plans for later today. And you put someone, let's say with autism in the scanner, and they're more likely to count the rivets inside the board. So they're doing fundamentally different tasks in the scanner, and maybe that's what we're actually measuring. And in one way that's, negative, because like you can just do a behavioral questionnaire, you don't need the scanner at all. But another way, if it gives us a sense of like, these are the networks that are changing and these are the networks that are engaging, fMRI might play a critical tool for us to understand, you know, essentially what their default brain states are different, and might split out these clinical conditions better. So I think we don't really know where this is going to go until we're monitoring what people think about while they're in the scanner.
Yeah, so because I mean this is really fraught. I mean, if
Yeah.
there was a reliable breakdown of the type of resting state that, you know, let's say, you know, some clinical population, if they always count rivets in the bore reliably, they do something that's similar. Great, then they will all group together. But otherwise, you're just going to get big scatter across. They all find some new thing to do. Some of them hum, you know, this first over and over the national anthem over and over. Some do the, you know, count the rivets in the bore. Some decide to count their toes forwards, backwards, forwards, whatever it is.
And there's two scenarios. One is it turns out that we're using this MRI and there's a great behavioral measure for what we need, and we save millions of dollars on clinical diagnosis. So that's a win. The other scenario is exactly what you're saying. There's a range of things people do, but by measuring that range and sort of accounting for their analyses, we might get, you know, closer to what the actual neural variations that matter are. And if you don't have some monitoring of what they're doing, are they counting their toes? Are they counting rivets? Are they planning about tomorrow? You just don't know.
You bring up a really interesting point, because there are many let's call them comorbidities, cofactors, just to be more clean about it. Because we know that, you know, with something like schizophrenia, there's also more of attention for real head motion.
Yeah.
Right. So you've got lots of other things going on as well. So if you can account for real head motion, then now you're talking about a sort of a next level down of, you know, in the data anyway, and another variability that's in the real brain networks. But what about, okay, going back to sort of, you know, knowing what the person's doing, like when most of us put people in the scanner, lights go down, projector goes on, we check email, do whatever we're doing, whatever it is. Rarely do we sit and glue our attention to a bore camera or anything else. And I know that there've been stories of, certainly in adolescence, where the kid was exploring the bore with his feet, you know, pushing his foot right. So that's pretty extreme. But we have real head motion and we have real other motion, fidgeting, which may or may not be reported in the real head motion, because it may be changing the loading in the coil, maybe just be, through space motion effects. How do we capture that? Are you thinking we should video?
I mean, this is like the whole idea of practical versus realistic. Like, if you want to set up a 3D video of their whole body and their face in the scanner, the tools exist for that, but that's just not going to go everywhere. But saying, hey, after a scan, do this 5-minute questionnaire, and it's going to sort of capture key data, that's easy. And we can argue what that questionnaire is, but it's much.
If you know you're studying a vulnerable population, population that's not a population that is vulnerable to being more fidgety, adolescents, you know, whatever. So
there maybe the camera becomes critical there.
Yeah. And wouldn't it be? I mean, again, this is just data. It's ultimately just 2D data. Aren't there, you know, methods that you could sort of basically, you know, throw this into a thing and say, give me an average motion index of the fidgetiness of the limbs that come out of it?
I mean, I've seen some stuff like that, and it's actually moderately, just to see motion is easy because in theory nothing moves.
Right.
So you just essentially take the first derivative of your movie and you get a decent first level measure of like how much is moving.
Right.
If you want to say like how much are the feet moving versus the fingers, then you're talking about like machine learning classification.
Sure. But if you just want a first level, because right now we're just putting in and we're assuming everything is stationary, which is probably, and we're not paying any attention to it. So if you have a vulnerable population and we record that data, then at some point, if you need to, you could go back and say, I've got an outlier here. What the hell is this kid doing? Or what's this person doing? Speed review the data or put it into a number crunch. Give me that first derivative number. Oh, holy cow. Lots of things were changing. Okay, we better go back and review. And you find, oh, look, the kid was exploring the ball with his foot. Okay, well, that's cool. That sounds like nice to have, and probably critical for certain types of studies, study specific, as you said. Anything else in the scanner? The questionnaire before and after, that'd be great to standardize
Yeah.
if
Yeah.
people find it useful.
I don't think anything else. I mean, I think the other thing is just sort of thinking what you're trying to, I mean, this kind of as a broad topic, like what is your task and what is your goal?
Right.
So like, do you actually want rest? Do you want a movie? Do you want a task? And I feel like it's funny, like the field started with like, really hyper specified task designs. And then rest was like, we don't need any of that. And now I feel like we don't want to go all the way back to like everything being hyper specified because that had problems. But it feels like the field is still trying to sort of find the right
Because
balance.
rest is ill-defined.
Yeah, absolutely.
As you've just pointed out.
Yeah.
Radically ill-defined. And that's potentially a problem. It's going to produce an awful lot of scatter. It's not necessarily the case that even the same person will do the same, will have somewhat similar resting networks across days, weeks, months, right? Because it depends on, they're getting divorced, their kids are, interviewing for college, whatever it is, So lots of things could change without you necessarily even knowing it unless you're capturing that information. Talking of capturing that kind of information, what are your recommendations on things like sort of capturing sort of your typical, let's call them diurnal effects, things that might vary depending on when the time of day of the scan, whether they've had caffeine or not recently, the kinds of things that can easily attention, right? Because after lunch, much harder than before lunch. What kinds of things do you think should be standard as just being collected? Blood pressure, sleep history, caffeine history?
I mean, I'll sort of in full disclosure say I'm bad at this stuff. So like I don't collect the one, like I can tell you, I can give you all these ideas and I probably don't collect them.
Right. No, that's we're just trying to define what people should be doing, and then it's up to them if they exclude them consciously. That's fine. I have no problem with that.
But I don't want to sort of talk on high, like this is what you must do, and I don't do that. But I mean, we definitely record time of day, obviously, because we know when the scans are happening, and that is stored.
And you probably try to control for time of day anyway, at least in certain studies, right?
Yeah.
You want things to be reasonably
the way we scan at where I am is we have fixed scan times that are just sort of pre-scheduled. So like
so somebody ends up with the 2 o'clock in the afternoon slot no matter what.
Yes.
So they're just doing sleep studies.
One group has 2 o'clock on every Thursday. So and that has benefits and problems. I mean, the benefit is it gives you stability both in like scheduling and time of day. But the mine is if 2 o'clock happens to be the really bad time for whatever your study is looking for. That's
yeah.
But
wow.
I really should probably be collecting caffeine and food status. I think those are useful, but I don't usually do that.
Right.
I mean, we can
just as a questionnaire.
What?
Just as a questionnaire, just a self-report. How much caffeine do you normally drink? Did you drink coffee this morning? How many cups?
Yeah, I mean, blood pressure, I feel like it's one of those things like I don't know how I would use it.
Right.
So it might be one of those things that 5 years from now, we're like, oh, now we know how to use resting blood pressure. And we should have been collecting it.
Right.
But right now I wouldn't, have a good sense of how I would add blood pressure to my analyses. Right. I guess that's really what comes up to a lot of these, like caffeine. I know caffeine's going to change things, but is it going to change how I analyze my data or how I interpret my data? And
no, probably not. Well, caffeine is a funny one, because it's so common. Yeah, and as I forget the paper, I'll dig it out, put it in the notes. But there was that study that showed that you want people to be on their normal routine. So if you're a 2 cup a day person like I am, don't come in caffeine naive, because then you'll actually have an abnormal situation. If you're habituated to caffeine, do the normal thing, right? That's actually better than varying the condition and trying to make it so that everybody's no caffeine on the morning of the scan.
But I guess my overall answer is like it's nice to collect this info, but I don't know if I'd use it in my analyses.
Yeah.
So
no, I think
actually the point I would use it actually is if someone's an extreme case. So
exactly. Testing
but that's tricky. Exactly. So like you're collecting it from everyone, and then you find that one person has 10 cups a day and drank like a huge cup of caffeine right before the study, and then you gotta
So the
say,
only, okay, so here's where I think it might be useful though, is whenever you're doing anything longitudinal,
Yeah.
especially if there's an intervention in that longitudinal study, even if there isn't,
yeah,
if it's just an observation, people getting older. But if you're doing any kind of intervention, any kind of therapy, whether even with, maybe especially if it's something like for PTSD or depression, anything that can change behavior, can potentially change people's habits, can lead to effects like changing caffeine consumption habits because they want to sleep more or whatever it is. You would like to try to capture that, I think.
Okay, there, I'll agree 100%. Yeah.
Yeah.
I say anything longitudinal.
Yeah.
If you think these might change, then collect them. But
Yeah.
for a
Or even,
single study,
or just assume they will change, measure them
Yeah.
anyway, and prove that they don't.
Yeah.
Because, eventually over time, people eventually do change. We get older, we have to drink less because, hangovers are longer. So eventually people's behavior does change. As you sleep less, you tend to want to compensate in other ways.
Yeah.
So I think that would be a good way to do it, or a good reason to do it. And the other thing, I suppose, is that just because I know it's your study in the first instance, but you've got to make these data publicly available.
Yeah.
Try to think like somebody else might
Yeah.
want to ask questions of your data. It's not a bad thing either. I think maybe more like a reviewer, just in case. If it's easy to collect, collect it.
I mean, if you're doing the quick, so then like if we're saying a quick survey is easy, caffeine,
Easy.
sleep the night before.
Yeah. Did you eat or
maybe
not?
last
What did you
meal.
eat? Yeah, exactly. Last meal. Yeah. How long ago? Roughly what it was it? Yeah. Quick, fast, really super quick. Exactly. Basic information. I would also include in that, I have recent exercise in one of my lists of things. So you want to know if they just sprinted, in the heat of the day, sprinted from the parking lot because they were 10 minutes late. You'd like to know that.
Yeah.
Right. Or if they just came from the gym where they did an hour of, spinning class, you'd like to know that. Cool. All right. I'd love to know hematocrit too, but I realize that's more difficult. Although with so many people collecting genotype, wanting genotype information now with those little sticky pad things, the blood draw things that you can put in the mail, hopefully those numbers will start to become available anyway.
The other one that Emily Jacobs has convinced me on, which I have yet to do, is all the endogenous hormones. I think is possibly even more important than hematocrit, just because we know testosterone, estradiol, all that stuff is absolutely changing stuff.
Can you get that out of just a little blood draw with these little sticky pad things that does the slow blood draw that you can put in the mail?
I don't know. I know that some of them you can get out of spit tests. Some of them you can't. Blood draws are more accurate. So it's one of those things that's on my list of like, yeah, we probably should, but I haven't actually started collecting those yet. And again, I think I agree with your comment earlier that this is much more important for like multi-session studies than it is for bringing someone in once.
Yes, right. Anything longitudinal for sure. All right, I'll look into that and see if I can figure that out. It'd be nice if it did come from one of these little blood patches because those things are so convenient. You can do it at home. It doesn't have to be contemporaneous with the scan. I can do it at home and a couple of minutes, drop it in the mail. And if we could get the hormones as well as the phenotypes, hematocrit would be nice, but not critical. Unless it's your study is studying people changing altitudes, in which case you need to know that. Otherwise, it's probably not super critical. It's probably a very small secondary effect. All right. Okay. Well, that's a pretty good list. We've gone through a lot of things there. So that I guess those are in the nice to have camp right now. Hopefully, eventually become essential to know phenotypes and hormone levels. That means we're going to be subdividing our data into more nicer, neater slices, which I like. Our groups, our large N groups, will be coming into smaller subgroups. Okay. Any other thoughts on stuff in scanning before we go back to your main list?
Yeah, I mean, I guess on my list, that kind of what transitions into this is more and more, I feel like things like all these physiological artifacts, we have decent methods to remove them, but they're never going to be perfect. And then we get things like heart rate correlates with attention. So there's really no way to perfectly separate something. So I feel like, particularly for clinical populations, that the path forward is designing our tasks that, and are not just tasks, the analyses that are robust to physiological noise. So, a resting state
Yeah.
scan where you're just measuring correlations across regions
Yeah.
is you can't really separate physiological noise from that.
Yeah.
But if you're correlated to behavioral changes, if you're, doing some sort of internal relationships to sort of, see regional differences, then you have a better chance of, identifying what an effect you're saying. I mean, the end goal is like, I'm seeing an effect. I am reasonably confident that this is not physiological noise because of something.
Yeah, I was talking to your boss the other day, and this subject came up. I made this chapter I'm going to write for him for this book he's editing, and I'm going to lead with that oft heard refrain about not wanting to throw the baby out with the bathwater. And I'm just going to point out there is no baby in bathwater. That's an edge. We don't have edges. We have salad dressing, and even when you leave it on the side for a week, it separates, but only so much. And there's always the amount of overlap between these areas. Not only is it blurry, but it's changing. And I think that your heart rate example is a good one. To me, it's the first order question is don't beguile yourself into assuming that it's neurovascular coupling in the blah 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 network when Occam's razor suggests that it's actually much more likely to be pulsatility because other things are changing. But you can sort of start to ask those questions if you at least expect your data to be lying to you. You can at least say, okay, let me go to an area which shouldn't have anything to do with this and ask the same question: is there still explanatory power? If there is, then okay, I have to hold my hands up and say I can't conclude much. It's dust in my microscope.
But if you're expecting, the whole fun is like the default mode network is also the heavily vascularized network of the brain. So like if you're just looking at default mode, you're looking at a vascular network. But if you're saying this same event isn't happening in these other vascularized areas, or the effect we're seeing is between motor and V1, but not other places, that's less likely to be a vascular effect. So even if there's vascular noise, there's, if your analysis has sort of internal controls to say whatever type of signal vascularizing you're making, it wouldn't be showing this style effect.
And that's exactly the way I think that optimistic point. If you recognize that your data could be lying to you, this allows you to then cascade your thinking and say, okay, well, given that maybe I need to do another experiment. Let's try to make it so that the heart rate is doing the same kind of thing, and now put a second effect underneath to try to just manipulate these 3 or 4 nuclei, whatever it is, right? So have a big effect and then ride some task on the top to try to sort of probe just that thing. That's sophisticated thinking. That's the optimist's angle here, is just to say, oh, well, it's hard. Is to say, no, well, it is hard and it's complicated, but if you accept that there are these limitations, this also allows you to make significant advances that your colleagues are not likely to make because they're going to just do the lazy thinking.
Yeah, that was making me think. I have a fun data set. This is still a work in progress, but I'm having fun with this data set I collected. So I did movie viewing and rest, not really rest, with paced breathing, but I am doing essentially paced breathing with changes in depth and amplitude
during the movie
and during rest. So essentially with a shift in the paced breathing, I either can do 2 runs that are identical or 2 runs that are completely out of phase or out of phase.
Do people manage to stay on task for the breathing when the movie's playing, or do they lose the plot or pick up
they
the plot
mostly stay on? I mean,
okay.
I think perfectly following the breathing is hard, but they do a decent job, and we're monitoring.
And how are you telling them to do the paced breathing? Is it visual as well with the movie? So it's in,
so I have a little circle in the center of the screen that just gets bigger and smaller.
And they're supposed to phase lock to that and then continue to watch the movie. And what kind of okay, wow. Okay. How's their performance?
I mean, some of them are really good, some of them are not as good, but I feel like everyone does it and some people kind of forget about the breathing and come back to it. But
it's a lot of things. Task switching, right? Be a kind of a distraction task. Yeah.
But the data is okay quality. I mean, I think the way I could say it's okay quality is, so what I had the same pace breathing, for a movie and rest run. So if they're doing it well, and you do sort of voxelized inter-run correlation, like inter-subject correlation, I see global connections between when they're watching the movie and just doing the paced breathing, because you're essentially getting the paced breathing
And
pattern.
you of course have those transitions where they go from doing the breathing task while watching the movie to now they're transitioning to paying attention to the movie. That's an interesting block right there. And then when they come back on task, right, you know, there's another transition. So you've got lots. It's a rich data set. There could be some quite nice second order.
I have them doing the same movie with different breathing patterns, so that if I can separate this breathing pattern, you're going to get better correlation between the runs.
Yeah. And
I have the same breathing pattern to rest. So if I separate it, we shouldn't be seeing any correlation.
Yeah.
So it's a fun data set that I feel like there's a lot more I can do with it, but I'm hoping to get, you know, at least enough to be able to publish and share it in the nearest future.
And the rest, presumably, then people are, their breathing is also still somewhat indexed to what's going on in the movie. Every time it's a suspenseful part, I'm sure they're holding their breath.
Yeah, but because it's the exact same breathing pattern, there's a little bit of a stability. And I had fun. It's the breathing pattern. I actually do AM and FM modulation. So like there's these tasks where, like, you know, breathe fast, breathe slow. It's just very subtle. There's no point in it where you're going to say, hey, you told me to breathe differently, because just every breath is like 10 milliseconds shorter, and then it gets 10 milliseconds longer.
That's
so
cool. Have you ever thought about just trying to surreptitiously, Blaze and I talked about this, trying to surreptitiously change the breathing gases just in a small, you know, the whole environment, just in a small, you know, fraction of a percent.
That's what this should do. They're changing from I think I do 4-second breathing cycles to 6 seconds. So just
but then you wouldn't need to worry about, you would control, you could put in some very complicated waveform of just blowing in, you know, 1 to 2, 3% changes in oxygen, maybe up to 0.5% change in CO2. No one would ever know. You just blow it into the scanner, into the bore, and then have that ride on top. And then you wouldn't even need to have them do anything. They wouldn't even, they probably wouldn't even notice. Chances are they wouldn't notice. That would be really, really cool.
Yeah. So, but this is, I guess this is just what we have. But
Well,
the breathing gas is like I've wanted to use breathing gases for a while, but I've never really sat down and done it for both, like just getting the protocols approved issues.
Yeah, I can imagine. Well, if you're doing, you know, when it's constrained on your face and when you're doing big manipulations, then obviously the safety becomes really critical. But if you're doing it, you can imagine like putting an end plate. Imagine just putting an end plate in like with a manifold and just blowing gases. You know, it could be just, you know, basically hijacking your bore fan and having a version that, you know, that's neutral is pure air, and then feeding it bleeding into that a few percent oxygen, you know, extra oxygen, or a couple of percent, you know, CO2. Probably wouldn't be particularly hard to get past the IRB, and I don't think the engineering would be particularly complicated.
Yeah, not too hard.
I'll look at that. That's the kind of thing I like to play with too. I'll have a look at that. I don't have Rick anymore; he retired, but I can still put some plumbing together. We've still got some bottles of gas knocking around. Cool. So and you had a list of things that you had designing studies more robust to physiologic noise. So you've got that kind of thing. That's an intentional manipulation of one variable over, or two variables, one physiological, at least one physiological. What else do you think what people should be doing for more robustness to physiologic noise?
I mean, the thing that I probably have been spending most time over the last couple of years has sort of been multi-echo fMRI methods, which I feel like there's what excites me about them is sort of like the current methods do help, but it's a wide open area of research. I feel like people just aren't thinking about much, because what you're effectively getting is another dimension of data that's saying this bit of my signal is more likely to have T2 star. This bit of my signal is not. And our current methods use that information, but there's just so many ways to use that information. You know, like with just the respiratory and cardiac noise, there's some of those signals that might be T2 star weighted and some that might not be. But just saying that, like, here is a respiratory signal that is, phase shifts from the chest, and it's noise, and we know it's noise, and it's no T2 star. This is a depth signal that has T2 star, and this is what the T2 star breathing signal looks like. So we actually can characterize better what, this T2 star variation.
So your primary interest at the moment, from the way you've described it, is essentially sort of, motion-like things, which would be non-T2 star from real physiology, which would be neurovascular coupling plus obviously physiologic variation.
Even simple things like the scanner drift. We see scanner drift that is clearly not T2 star weighted.
Yeah.
But one of my colleagues, Jen Evans, had a paper a bunch of years ago which did a very slow checkerboard change and was able to distinguish, and that's essentially a drift. Yeah. And she was able to distinguish that drift as a T2 star signal from scanner drift. So suddenly that opens up possibilities for pharmacology and also other stuff, because you can see these slow changes over time and say that actually has a T2 star component, while something else is clearly not T2 star and more likely to be drift.
Yeah, because most people would just filter that out, right? They'd just do a high pass filter and that would just disappear. But if you were specifically looking for long-lived effect, because that's always been an Achilles heel, right, for fMRI or for BOLD, if you're looking for longish tens of minutes, things like sleep come to mind, right? Effective drugs over long times, then this would be another way to get at that. That's a good point. And then you would use this as a step. I think maybe the problem here with multi-echo is that it's maybe overpromised in the. I think people were thinking, oh, it'll get rid of physiologic noise. We'll just magically filter everything, and the only stuff that's left will be what I'm interested in. But obviously, physiologic noise is real T2 star, so that's still in there. And now you have
some is so like pulsation is not T2 star,
right?
Phase shift isn't T2 star. The depth breathing, but interesting.
Nor some inflow effects. That's real activation.
Yeah. Well, maybe depending. It's like we have a study that did gated fMRI, and that has massive inflow effects, and using multi-echo information.
Because the TR is varying quite a bit.
Yeah. Yeah. And the multi-echo information was able to essentially remove the gating effect.
Wow.
So.
But then isn't that also then essentially pushing your spatially pushing you towards the veins? Now, those inflow dominated voxels will be mostly capillaries and maybe small arterioles.
You mean for the gating or just in general? Or?
No, in general, anything that's inflow weighted will generally be on the sort of tissue or upstream side.
It's going to pull out like the T1 style inflow effects.
But sometimes you want those.
Yeah.
I mean, they go in the same direction as the T2 star, right? They're normally additive. Mm-hmm. And if they have better spatial specificity than the T2 star, then I guess it depends on what you're interested in.
I think what I would say to that is you have the data. You can distinguish that.
You could decide.
So it very well could be that, I mean, from what I've seen, it's not like I'm seeing something that looks like our task that's inflow that's not T2 star weighted, and something that looks like our task that is T2 star weighted. It's usually there's the stuff that's designated not T2 star. It does not look good. It's like it doesn't look like a thing.
So maybe there's another way to approach this, which would be something like to look at the spatial ICA or something like that. But some sort of how many voxels? What are the next nearest neighbors doing, and how much correlation across gaps, right? Because that cannot be neural, right? If it's 20 voxels away and it has the same, it co-varies.
That's actually brain. The main method actually, that's pretty much what it does. So it does ICA.
Okay.
And it has one measure, spatial ICA, and it has one measure, which is essentially how T2 star weighted or not is the component. And then it has other measures, which are like, for T2 star weighting, is it sort of white noise across the brain, or is it clustered? So they're measuring, it's like, is it looking like how we'd expect things to look?
And where are people learning about these kinds of the best practices for multi-echo? Are there,
Yeah.
are there papers sort of guiding people for this? Because I just noticed that the most recent psilocybin paper that came
Yeah.
out, the Washu
Mhm.
group,
Yeah.
Rosenbach's group, they did multi-echo, but they just
Yeah.
did the weighted average and then carried on. They didn't bother to exploit the multi-echoes
Yeah.
beyond just taking the SNR.
Yeah. So one of the things I work on is I developed this software called Tedana, Tedana. And as part of the documentation, we've been trying to compile just general education resources. So there's a lot of references and papers and talks.
Now,
there's
they're relatively well-established pipelines for people to follow to sort of get going on this.
Yeah, so I mean, like the software that I developed today, like, is pretty well established. I mean, I think this also goes to the hype question you brought up earlier. So multi-echo has been around for almost 30 years.
Right, but Prantik's paper was only
Yeah.
what, 15 years ago.
Yeah. So he published his, and so like it was around for a while. Then he published his, and there was a huge amount of hype. But I think the two reasons that hype died down were, one, his software, like he was a phenomenal grad student. He's still a phenomenal person, but one grad student writing code, that's just not robust usually. I mean, there's exceptions.
Yeah.
So it wasn't quite as push button as people to use. And the other issue was the vendors didn't really have the sequences. So it was kind of, special whips and things to get the sequences. So a bunch of people were like, this is the next greatest thing. They tried it. They either couldn't collect the data or they couldn't process the data, and they kind of gave up. And then about 5 or 6 years later, the vendors caught up and the software developing got more stable.
But
And
we've
now
got, like, I mean, on Siemens scans, you've got CMRR has multi-echo,
Yeah.
right? I mean, so everybody who has CMRR has that
Yes.
capability.
And that's exactly the point. So like in 2012, that didn't exist. And then
Right.
CMRR added multi-echo. Like, I think it became widely used in like 2016 or 2017. So,
But even so, like, look at Dosmark's recent paper.
Yeah.
They're collecting the data. They did 5 echoes as well, by the
Yeah.
way, a lot.
Mhm.
So they did, that's yeah. So, but they still just did the brute force
Yeah.
processing. Seems to me that the hype. I remember
Yeah.
thinking people would expect to get, bold equals what I want to see, non-bold equals not what I don't want to see, and now people like you and me keep bringing up physiology,
Yeah.
and they're like, God damn it, it doesn't separate like that. Again, it's salad dressing, not babies in bathwater. So that's probably some reluctance. But is there a good way for someone to sort of get going and do the spatial ICA, ask to practice with reasonable data sets to sort of get comfortable with interpreting what they get out of this stuff? Because a lot of this stuff is black boxed.
Yeah. I mean, I guess I'll share one thing. So like with our software, like our software is pretty, like, there are issues. I can, if anyone has issues, I can talk about that nonstop, but it's fairly robust and fairly stable. And one of the very explicit things we did, let me see if sharing works.
Did I change it?
Yeah, I think I got it now.
Yeah, there we go. Brilliant.
So when you run our results, every time you run data, we give it an interactive report. So this is a checkerboard task. Here is an accepted component. Here is the time series of my block design checkerboard. Here's the map. And this, you can hover over it. This kappa is kind of the T2 star weighting, and rho is the S naught weighting. So you can actually just inspect your data and look at it. The second thing, like the biggest rejected component on variance is linear drift, which is not surprising. But it means that once you collect your data and run the program, you actually get to look at it and see what this looks like. So this is not really bold weighted. This is very likely, she could be cardiac or respiration. It's hard to say, because it could be cardiac folded over, but it's clearly.
What's the kappa and rho there? Is that in the elbow? What does that wedge correspond to?
So this is a rejected component. It looks like it's below the elbow. So it's saying that it's the kappa elbow in this case. I have a vertical line here. So that's kappa elbow. It's below that elbow over here.
Okay.
So this is not really teach star weighted, and we could see it. So I think as far as getting started, we have a good community to help people learn how to use the program. And once you have it on your data, you can just look at it and get a sense of, this is probably cardiac.
And that's above the elbow?
It's above the kappa elbow, but also above the row elbow in this case.
Yeah.
So it has, and it's, yeah. But you can look
And
at
that,
this.
and that, yeah, so that, but it's a pulse, so but pulsatility at the heart rate, you'll have intravascular bold, you'll have extravascular bold that is being modulated by the pressure wave, at least on the arterial side.
Yeah.
The venous side, less so, but maybe a little bit, a tiny bit. And then you'll have the artifactual sort of CSF type motion. So there should probably be a couple of different components that will have somewhat similar spatial properties, but quite different kappa and rho values.
Absolutely. And one of the things that we're adding in very soon is you can correlate to the time series. So you can correlate, you can make a CSF time series, you can make head motion time series, and it'll give you for each of these components how it, how it fits to them. So whether or not you use them in your decision process, you can say, here's a rejected component that looks like it's highly correlated, it's highly fitting to head motion. Here's an accepted component that's really fitting to breathing.
Yeah.
So it, like I'm saying, like we have our current method, but if we're trying to understand like what the system and factors are, understanding what does a T2 star breathing component look like. I don't think we have a great sense of what the unique T2 star contribution to breathing looks like versus breathing artifacts in general. So this, if you're collecting multi-echo data, that gives you a chance to actually answer that question.
Yeah. The pulsatility is especially interesting, I think, because that's now you're starting to approach what ought to be our fundamental limit, right? Because at some point the brain is doing this, the vessels are doing this. Now the idea of rejecting things that are motion contaminated becomes, now again, there's salad dressing. You can't easily separate those two things. But if there's information in that, and that information is correlated with a task, if you at least can show that there's changing pulsatility, or there's some non-T2 star or T2 star change that's localized.
Yeah.
Do I need to be pure about whether it's bold or not? Probably not, right? It's
I mean, like we can
telling me
make
something about that part of the brain that's involved in this outcome.
Yeah, I mean, I think, in our code and in general, you can fit whatever you want to the models. So it can give you that information, and then we have like the rules that we're distributing, but you can make your own rules. So you might be, I think one way of saying is like, do you want to be conservative or not?
Right.
So do you want to say, I want to remove anything that might be noise? Or do you want to say, I never want to remove anything that might be neural? So you can tweak things to be, with either direction, and whatever you reject, it also gives you information on what's happening. So, and I'll just say, like, the method is not. I mean, I have ideas for sort of totally different ways to use the multi-echo, but I think what excites me is like you get this added bit of information, and we've barely sort of scratched the surface on like what you can use it for. And this, like, I see is just a really simple.
Yeah, right. Okay, so backing up a sec, assuming people are like, oh shit, yeah, I got a good move to this. I got a how many TEs? Any other caveats on the acquisition side before we get too carried away about the acquisition, processing?
So I would say it's pretty easy to get. So if you're fitting data to a curve, you need at least three.
At least three.
And it's pretty easy to get three. Because what I generally say is you have your voxel size, you have your coverage, you have your TR, and you have acceleration. To go from one to your first echo is pretty much free because it's essentially you're collecting in dead time. Your second echo is around what you would normally collect. So it's really that third echo that adds something. So, if you increase in slice acceleration, I know you have opinions on that, by one factor, then you essentially get the three echoes for free. if you increase your TR a little bit, you get it for free. If you, cut off a few slices, increase your TR, you get it for free. So you can get that third echo relatively cheaply, and that weight, and like you were talking about the weighted average earlier. So if let's say you increase in-slice accelerations, you go from no in-slice acceleration to 2. So that knocks down your SNR by square root of 2. But now you're collecting 3 echoes, which means you're averaging 3 times and you're increasing your SNR by approximately square root of 3. So ignore it, and you have a shorter readout, so you have less distortion to a certain extent. So to get 3 echoes takes a little bit of work, but it's close to free. When you go beyond 3 echoes, then you have to make usually you have to make some compromise on something. So you might have to push acceleration a bit more. You might have to say, maybe I don't need the whole brain or something, but it's doable.
Yeah, I was going to go up. And is there a big benefit to getting to 4 or 5? 5, I can imagine.
Yeah,
having quite a lot of benefit.
Yeah. I would say right now, with the current methods, no.
Okay.
But if you're collecting a data set that you want to be using for years, I wouldn't be surprised if there's a method longer out where 5 is useful. There's also, the other thing that I haven't had a chance to use yet, but there's this time resolved EPI sequence, EPTI, which by effectively the way you could use it is it's kind of like a sliding average of echoes. So you can get an echo every few milliseconds. And they're overlapping. So like, your 5 millisecond echo contains almost the same data as your 10 millisecond echo, but you get a full curve.
Yeah.
So I think that I'm very curious. There's one paper that's been using that with multi-echo denoising, and I'm very curious to see where it goes. And then similarly with some of these, GE now has like a head only.
Have you read those papers? I tried to read. I tried to read those APTI papers, and most of it went over my head.
Yeah.
What's the downside? You never get something for free. You're doing that EPTI coverage. Is that a 3D volume, and it's slow 3D sampling?
I think it could be 3D. I think you could do 3D or 2D if I remember correctly.
But if you're doing 2D, you can't move on to the next slice. You can do simultaneous slices, maybe.
Yeah.
But again, it
Maybe
seems like
I'm trying to remember.
When I look at that, I seem to think that there was something got slow, right? Because you never get something for nothing. Something got slow, and I thought it was the repetition time. I think the minimum repetition time is quite long.
I think that it's kind of like folded in acceleration, so that like within an X second window, you're not collecting as much data, but you can sort of average things over. So there's a bit of like a dependence across time. If I remember correctly, I haven't
Maybe blurring. Yeah, so I'll have to get Larry or
haven't read it in a
those guys
while,
from MGH to
but
come and talk about that, because I have questions. I don't
on the
fully
plus
understand.
side, it dropped distortion because of the way it's set up. You have extremely low distortions. So if you have a sequence where you're really worried, if you're really worried about distortions, it's extremely low.
Yeah,
So
okay. I'll have to go back and look at that, because I seem to remember that, because it's like the Heisenberg's uncertainty principle.
Yeah.
You want to know more with high precision and no distortion. Something else gets bigger. I think it was the repetition time, but the effective repetition time, not the MR repetition time. The time required to get your volumes worth of data. I'll go back and check that, and I'll put the references down. That's a good point. So, all right, well, 3 echoes. People can do that without breaking the bank, it sounds like. Anything else on multi-echo that you want to encourage people to be doing? It sounds like people should be playing with these toys. It sounds like this is an area that hasn't been poked at much for the last decade.
I think, there's definitely, I think from the analysis side, there's a lot of open space of what could be done, and I'm happy to talk to more people offline if they want to sort of work on the method space as well.
All right. All right. What else? You mentioned the notorious GSR.
Yeah. Okay. Yeah. Global signal regression has been a headache for a while, and it was really interesting at the last HBM. There was a session on global signal, and I was dreading going to it because every time I go to one of these, it just, my blood pressure spikes. But there, I mean, number one, there's some very good speakers in it, and the thing that really jumped out at me, like there was talks about, like, you know, when to use it, how to use it, but no one in the session was talking about GSR as a physiological noise removal tool. And it just sort of clicked that, like, if you think about, no, so if you think about, like, when people talk about it, like, why is it done? It gives us these pretty maps with really crisp, positive.
Oh, you mean they just don't realize that the reason why they're doing it is because it's actually doing a physiologic noise removal with zero lag.
It's doing a few things, but I think what's interesting is if people talk about sort of removing variation across the study, they're talking about, you know, a really crisp positive and negative network. It's, you know, there is noise removal built into it, but I'm realizing the way people talk about it now, it's much more of a data transform step. So you put it much more in, you know, the space of, like, aligning to a common atlas. Or when we're doing machine learning, it's data regularization. So data regularization is you shift your data set around the mean of 0 and, you know, and give it other properties. That is literally what global signal regression does. And if you look at a lot of the analyses where it's been successful, it's analyses where you want to sort of shift your mean to around 0 so you can better compare across the population. So I think
that's the argument being statistical in the negative correlation issue notwithstanding.
Yeah, so I think
interesting.
If you're framing, and I actually sort of have a half-written review paper, opinion paper on this, so we'll see what comes out of that. But my sense is, like, yes, it does remove some physiological noise, but we also know it removes a massive amount of neural activity. But if you think about it as, like, is it removing attention? Absolutely, it's removing an attention effect. So are we saying that's noise removal? No, attention is not noise. Attention is neural. But if you're saying, I want to normalize, I want to do a data transform and normalize for attention, then maybe it's an okay step, but it's a different framework. So then the question isn't, like, do I do global signal regression? It's, how do I normalize for attention in my analysis? And maybe it's the right step, maybe it's not, but you're shifting the question. And I think if you frame it as a data transform, then you're much more you're comparing it to the other data transform steps. You're not comparing it to the other physiological noise removal steps. And I think that's where it should be. And you know, in my cynical thing, I think if you compare it to the other ways you transform data, it's still gonna be a bad choice in a lot of cases. So I'm still sort of an anti-global signal, but I think it's
you'd want to
conversation.
Yeah, separate the parts. You try to get get everything in one step, which doesn't doesn't make any sense. That's kind of a miracle.
Yeah.
But yeah, if you did, like, you know, rapid tide correction, which is essentially, you know, a lag appropriate physiologic correction, which starts with the global signal anyway, from the Erdogan 2016 paper, then if you take out anything that clearly flows with the blood, right, which is then at that point, now you could go back and ask, what's left, what does the global signal look like now?
Yeah.
Right. And now maybe you can make some more logical decisions about zero mean and recentering, etc. Maybe that now makes sense.
Yeah, but I mean, push comes to shove, like, we know with absolute certainty there's a lot of neural activity in the global signal.
Yeah,
there's been enough studies that have shown this in enough different ways that, you know, if you remove physiological noise, that means it's going to be even more neural. So
yeah, so it'd actually be worth then throwing away the new global signal, the corrected global signal. You should definitely not want to throw away, because now you're definitely throwing away attention and everything. Wow. Okay. Yeah. So either way, don't do it.
Yeah, I mean, like, for the papers I've collaborated on, the ones where I think it's been useful is essentially when you're doing regularization. So if you're essentially turning your correlation matrices, if you're making correlation matrices and you're z-scoring them, which is a common step in a lot of analyses, that's effectively global signal regression. And now then you can then ask, like, why don't you just z-score? And the answer might be no, z-scoring is easier than global regression, but, you know, mathematically they're extremely similar. So is it bad then? No worse than z-scoring, but you have to decide, like, is this an analysis where you want to z-score your connectivity matrices? And if the answer is yes, then you do global signal, it's no big deal, but why not just z-score?
Yes, right. Interesting. Okay, well, okay, and you've got a review paper you're going to put out on that?
It's yeah, we'll see when it comes together. It's after the session at HBM, I sort of had like an inspired evening of like writing a page or two, and now it's sitting. So we'll see
Well, Peter's got that challenges, fMRI challenges, which I think he's going to do as an edited book now.
Mhm.
So use of global signal regression, that could be a good
Yeah.
challenge article. Maybe put it as a chapter. There you go.
We'll see.
Right.
The trouble is every single opinion and piece on global signal just causes so much fighting. So so we'll see. But we'll
Well, okay, yeah, but that's in a way that makes your case, because
Yeah.
if there was a clear answer, then it would, I think the problem is people get religion, right? Because it depends on their perspective, what they're trying to do with it, trying to get out of it. And there are no negative consequences, there are no personal negative consequences to being wrong or whatever, right?
Yeah.
Nobody dies. So you can afford to be strongly opinionated, even if the thing turns out to be fundamentally flawed. All right. So any other thoughts on GSO? You want to move to down your list?
Yeah, we can move down the list.
So biases and noise sources, people think do or don't matter.
Yeah. I feel this kind of related, this kind of also opened an idea for me. Like I know you are very focused on like respiratory and cardiac and physiological noise. And in an earlier discussion on global signal, actually, there was someone who said, like, a senior researcher who is very invested in using global regression was like, motion is, especially in populations, is the most important artifact. And it destroyed this result I had once, and we need to do everything we can, censoring global signal, to get rid of motion, because that was it. And the truth is, of course, it's both. Like motion causes trouble depending on your analysis. Respiration and cardiac seriously causes trouble in a lot of scenarios. But it was just sort of eye-opening that we were having this argument about global signal, and the argument was actually, is motion more important than like I was arguing like retro ICOR, RVT, rapid type stuff, and he was arguing global signal, and that wasn't actually the argument. The argument was, do I care more about head motion or do I care more about breathing? And it was a fascinating argument, because I hadn't thought about it that way before.
Yeah.
But with hindsight, it kind of shows that sometimes the arguments aren't what you think they are. And in the end, like, if you say like, no, we got to remove both and we got to figure out balanced methods, I think that's the path forward. But people do have different perspectives on like what the artifacts are that matter to them most.
So my perspective is that I'm actually becoming less interested in the sorry, I'm cramping my leg. I'm actually less interested in the consequences per se, whether people do global signal regression or whatever.
Yeah.
I'm trying to look at this as like you're studying a whole organism instead of brain floating in a vat. You've got to, there's all these feed forward and feedback mechanisms. We probably can rank the most serious issues, depending if you know a lot about the population. But for most modern interesting studies in fMRI, things like psychedelics, you can't separate these parts, and you probably can't even rank them. Because everything is going to potentially change. So my role right now, I feel like, is just to flag all the factors. And I don't even like the term noise. It's not noise, it's signal. These are information carrying temporal variations, and they may be big, they may be small. You may want to rank them, you may be interested in ranking them. That's fine. I would just like to make sure that people are aware of all of the ways in which their data may contain information, such that if they're bothered, they can try to pull it out. And I'm not at all worried about thermal noise, true noise, right? It'd be nice to get to that point. I don't think we'll ever worry about that. I think we're in a motion limiting regime, and when we're not, we're in a physiologically limited regime. And then we are probably limited by the biology, because you can't, like, at some point, motion is a neural process.
Yes.
And it's going to have a okay, you could, I was thinking this morning, actually, one of the ways I can artificially separate these things is to think about them as subcortical, breathing, autonomic stuff, and cortical.
Mhm.
But again, that's not a hard edge. These
Yeah.
things talk to each other, right? So, we have a category, we have a lazy thinking problem. Maybe that's closer to why people argue about this stuff, because they've got to get results out.
I mean, I think I'm very much aligned with you here. It's just that it's a system,
Yeah.
and when you hyper focus on one part of the system too much, you're kind of missing the you sort of drift away from the actual question.
Yeah. And unfortunately, sometimes you can't separate the parts. There's no way to do it in principle. So, just have to figure out, get as much information as you can, and then ask qualified questions, and realize that the answer may not be, the mechanism that you propose may not be the one that actually explains the data, which is fine, just as long as you keep an open mind, and don't just try and push a particular viewpoint. Well, cool. Well, that's why you do another experiment, right? Because then if you do a good experiment, then a new hypothesis should emerge, and then you should be able to push on that hypothesis to try to figure out whether the mechanism was this or that. Right? If it's respiration, you try to break it with a respiratory task or some physiological modulation. I did like that. And again, go back to the Dosenbach paper. One of the things, I haven't fully read it because it's huge, but I did skim through the supplemental and looked. I liked the fact that they thought to find a control drug that had similar physiological phenomena as the psilocybin they were trying to study. So they got cardiac receptors, etc. Huh?
That was a part of the paper I jumped to too, because I think we think similar, but like
Yeah,
it wasn't actually. They said it should be similar, but then they collected data, which is good.
Yeah,
and it wasn't similar.
And it wasn't similar, and they didn't talk about it. No, I agree. So that's where I'd be starting and digging and asking the aggressive questions. But at least they thought to ask that
absolutely.
Question, and the data exists, which means that again, thinking like a good modern citizen, is that now somebody can come along behind and poke at those data.
Yeah, absolutely.
Ask those questions, which is great. Does the heart rate variability, is it really the same? And are there other ways we can get at these same data, which I think was super cool. And a multi-echo, so someone could also do analyze the multi-echo. Well, cool. So do you have any other words of wisdom for biases in noise sources, or just to try and encourage people to keep an open mind?
I mean, I think the biggest thing is people have to stay close to the data. I know you talked with Joe Etzel a bit about the quality control project. I think the biggest eye-opening part of that is, I mean, this was not really eye-opening to me, that just because someone shares data does not mean it's good data. And the number of times I've heard, like, I've analyzed, for my study, I've used the gold standard Human Connectome Project data. You could think that data is the best data in the world, and it still has problems. It still has serious problems because it's real data in a real biological system. And the only way you find out what's in your data is looking at it. And there are some pipelines out there that are very much I feel like there's this sort of push and pull on, we want reproducible, reliable pipelines, which means you push a button and you analyze your data. And there's a benefit for that, because people push the wrong button and they get different results and it's a mess and they can't reproduce. So, like, that's an actual issue.
Yeah.
But if you push a button and get your end stage, you're not going to see anything in between.
Yeah.
And I think that's, more than any specific issue in the data, the fact that people are getting used to not looking at intermediate steps and looking at, did my alignment work? I've done something for noise removal. Did the noise removal actually do what I expected it to do? Is in some ways more.
There should be flags or scores should come out at each step, at least at the pipeline, and say, traffic light, green, red, yellow. Yeah. Right.
And I mean, I am biased because the AFNI team is down the hall from me, but this is what they've done, and they really try to make it possible to sort of check every step along the way. This is
Are the checks done? Are they just sanity checks that tell you, say green, carry on, or do you have to look at something and make a judgment yourself? This is correct.
They do both. So
Okay.
you run a script and it runs your entire pipeline without stopping. So you get that done, but then it outputs sort of summary statistics in a report. And the report, like this is what your dropout looks like in your epi image. This is how well your epi aligned to your anatomical. This is how well your anatomical aligned to your template brain. This is looking for various types of artifacts in your epi time series. This is what your, did your head motion, here's numbers on did your head motion correlate to your task design. So they try, I mean, they keep adding to it, but they essentially, these are measures, as many summary metrics at every step of things to look out for.
And then what
And
do people do? Do you have like some sort of error bars? You say, well, if it's within,
yeah,
if it's above a threshold, keep it. Otherwise, put it in the.
so that I'd say that's left to the user, but there's a few things like you can set thresholds. So like for head motion, you can say, censoring, you're censoring above a certain point,
Right.
or you can say,
which all on its own needs
yeah,
some caution, because you have to think
Yeah.
about the consequences of censoring your data. Does that even make sense
Yeah.
in certain situations? Can you have gaps in your data that aren't going to cause issues?
It depends on your analysis. So I think that's
Exactly. Like, so if you're like you're free thinking, if you're going to do a free thinking, resting state, then gaps are going to be a problem.
Yeah.
Right.
But it's also like, you can set a threshold saying before you look at it, if more than 10% of the volumes or 20% of the volumes are censored, it's done. You can't use this data.
Right.
But then you have subtle things where like you have, let's say 5% of the volumes are censored, but you can see that your motion time series are causing serious artifacts. So I think you can't make hard rules, but if you go through step by step and you say, there's not as much motion, I've also seen things where like there's not much head motion, but the head motion is task correlated. So having something that says like, what's
Because of chest motion, because it's pseudo motion.
yeah.
Yeah.
So just having that and saying like, whoa, it's not much head motion, but I look at my task and it appears in the edge of the brain and the ventricles because it's correlated to
I made that point in a blog post recently because I was reading one of Joe's posts, and I was just thinking like, including pseudo motion and real head motion in the same frame-wise displacement doesn't seem to make any sense to me if you can separate them. And it seems like because the pseudo motion is a translation, I would probably try to do a separate pitch roll your frame-wise displacement, because that should be capturing real head motion mostly.
Yeah.
And then maybe there's a sort of secondary analysis you could do, because that would, in that particular case, you would think that it would say, well, this is pseudo motion. You've got a lot of pseudo motion.
Although going back to multi-echo, I don't know if you saw this paper from Tom Liu's group recently on they did multi-echo data, and the standard sort of like 28 millisecond 3 Tesla echo is head motion correlated to global signal. And I think you probably get more of those artifacts because you get a bit of T2 star signal that warps your motion parameters. But if you go to the
because
first
because of the shim, just because this is real head motion,
yes,
causing real shim issues.
Or it might be pseudo head motion. It might be pseudo head motion with T2 star, because if you go
that's
to
true.
the earlier,
That's true. Yeah, because it's causing nonlinear effects through the field.
the earlier echo doesn't have that effect as much.
Yeah.
So I'm curious, like I haven't looked at this sort of with the pseudo motion that Joe talks about, but I'm curious if the pseudo motion is also a little bit less in that earlier echo. And again, like with the multi-echo stuff, like this is also a possibility of separating them out. So if we're collecting multi-echo, particularly multi-echo with phase, it might be possible to like 100% identify what is pseudo motion and remove that, not even include that in your motion correction translation. Just
yeah. Well, and it should follow certain spatial rules. Are you doing spatial action? So it should follow the field direction. It should follow there should be some spatial criteria that you can impose, or at least ask the question.
Yeah.
Does this make sense from a first order, second order spatial effect in T2 star in the shim field? Yeah.
But this is one of those things that like, this isn't like the denoising methods of multi-echo, but like that first echo has different weightings that might affect how we calculate head motion. And that's a totally.
Yeah. Wow. Wow. Okay. Well, that's another good reason to do multi-echo. Yeah, I just sort of, yeah, I mean, the whole pseudo motion thing, I think we're all aware of it sort of
Yeah.
now, but yeah, we don't really, we haven't done anything with it. The fact that
Yeah.
we've recognized the problem, we're still
Yeah.
lumping real motion and pseudo motion in frame-wise displacement, and
Yeah.
we don't have, we're still assuming our volume realignment is fixing all of the sources.
Yeah.
And we haven't really asked him more sophisticated questions. That's a good point.
Yeah.
Well, there you go. There's another area ripe for some some new work too, to try to move things forward. So maybe multi-echo there. Cool. All right. Well, what else? Any other? How long have we been on? Hour and 20?
Yeah.
Cool. Well, time flies when we're
Yeah.
ripping through the. What any other thoughts that you want to
Yeah.
get out there into the public domain?
I mean, I think the other thing I would just sort of think about a while that relates to some of this is we've had this big growth in data sharing, and I think that's good for the field. But I feel like the growth in data sharing combined with the pandemic means that like we have a lot of people entering the field who have never collected data. And all this stuff on what good data looks like and good bad data looks like. I mean, I could be the grumpy old guy saying kids these days, but you know, it's not their fault that data collection is expensive, and it's not fault that a pandemic shut down data collection for a couple years at formative training years. So it's not really, you know, I guess really it's like the grad students and postdocs right now that there's a whole generation that really didn't get that experience. And
Well, and on top of that is the fact that you can get, it's not getting any cheaper. And as we were talking about before we came online, you know, about grants and whatnot, people retiring, it's not their fault if their boss's grant didn't get renewed or they can't get funding because scanning's still expensive and you get free data off a database.
But I feel like there has to be, I would love to say everyone should collect data at some point, but I'm not sure that's realistic because like there's so much, there's a fundamental understanding you get from collecting your own data.
Yeah.
But if that's not realistic, then the question is how do we train people who aren't collecting data on what good data should look like?
Yeah.
And the QC project that you know Joe mentioned and I'm mentioning now, I think is part of it, but sort of on a fundamental level, this just wasn't an issue. Like when I started, like everyone collected data, everyone saw more of the guts of the scanner than you hope you wanted to see.
Yeah.
And the idea that someone can get a PhD in cognitive neuroscience and never touch an MRI, never see where their data came from was just unimaginable 10 years ago. And now it's normal. And I feel like this change happens both gradually and fast, and people haven't really grappled with what that means for teaching.
Interesting. That overlaps with another thing that Peter and I are talking about, this idea of a core curriculum. To get people all on a common framework, because still there's a bias between people coming out of physics and psychology programs. I think there's probably somewhat similar biases as always been. So still some bioengineers, some biophysics, etc., but still probably most people coming from those programs. And now you're asking them to basically become an interdisciplinary expert in a dozen different fields, from biochemistry to physiology to statistics to.
I mean, the one thing I would also say, like, in addition, like, so NIMH does this summer course every year, which we've now been putting online on YouTube. So you can see a few of my lectures and others on that as well. But my vision to do for 1 year, which takes work, is essentially, so you're actually going to collect MRI data as like the whole course. So like there's a bunch of like intro MRI courses for like upper level undergraduates or graduate students, but the really practical of, okay, you're really collecting data, what do you need to think about? Maybe I'm just not at universities, but that doesn't seem to be a course I've seen around. And I feel
it's done much more vocationally. It's the people who are going to collect data, that's what they learn. Otherwise other people don't learn.
As far as the core curriculum, I feel like the other separation is no one needs to know everything. They need to know what they don't know.
But everybody needs to know the foundations. Like if you're going to, even if you're not going to collect your own data, if you don't understand the oxygen extraction fraction, you've got a problem. If you don't understand functional hyperemia, you've got a problem. If you can't understand, nobody would expect somebody coming into from a psychology program. It's expected that you know what the T statistic looks like and normal distributions and certain other things, but it's because that's their historic bias, just like physicists understand electromagnetism. But then when you start talking about respiratory physiology, well, where's that in everybody's core curriculum? It doesn't exist. So if we don't make it a core, if we don't have, and we're talking about sort of common things. I mean, if you going back to the data collection, so if people are not, let's say that there is a way that you could sort of give people courses to get them at least the experience of some of the experience of collecting their own data, even if they're not going to do their own study via, let's say, a summer program, or maybe it's online and they're watching it through Zoom or whatever. Do you think that they would need the foundations first to be able to understand what's going on into the data collection, or do you think it's its own set of issues? It's like bench chemistry versus chemistry. So I was trained as a chemist, right? You can know all of the theory, you can push electrons around on a whiteboard all you like, but when you actually come to measure shit and do things, right? It's completely different.
I think that's the hardest to teach artificially. I think is the trouble. You collect a data set and learning. I mean, what you really want to do is learn how to say that something looks weird, and that's the hardest thing to teach, unless it's like part of a project. Because you can't just sort of give someone a data set and say, "Tell me what looks weird." They have to be invested in the data set. They have to sort of have a reason to care about it.
So we kind of need a new teaching framework, because clearly, the number of scanning centers is likely to go down eventually because funding will become trickier. So, or at least the ratio of number of people doing fMRI to the number of people scanning will go up, right?
Yeah, we're going to keep getting people
to get people on the coalface. What
There's
does the theoretical?
Otherwise,
we should look at physics. I mean, physics divides between theoretical and experimental physics, right? But they talk to each other, sort of, right? But presumably they have at least an appreciation for each other's domains. There must be some kind of cross-training going on. You can't just be pure theoretical without knowing fundamentally what the limits are of your experimental apparatus, right? And presumably vice versa. You have to have a theory before you can go chasing experimental data. So maybe I'll look into that too. Maybe there's a way that we can devise a new kind of experience of collecting data, that a teachable course that maybe maybe the same data can be reused. We don't have to collect new data in real time. Right. Maybe there's a way that you can hack a virtual practical course.
But so that's been on my mind recently, because just it literally is a whole generation of people who don't really know what issues of data look like, because they were handed data sets that other people told them they look good. You need that training. Like it's not just something that magically, you can't learn it from a book, at least not an existing book.
Well, right. Where I mean, the part where I'm struggling right now, I mean, I could imagine like you could create like a virtual scanner. You could have virtual data, and could be quite realistic. But you would still not necessarily have the part that might make the biggest difference, which might be the way that you interact with the subject. Right. So it might be that your bedside manner is affecting their anxiety and affecting that and making them a better or worse subject, right? And that might be something that you're not aware of because it's a virtual scanner.
I mean, that's like my ideal is everyone collects some data, but I don't want to sort of get that ideal in the way of the reality of the current world. But absolutely, the ideal is, there's this push against, n equals 20 studies, and I think it's a bit overblown. But the benefit of everyone being involved in at least one study, I think, is huge. But even that, I'm not sure it's realistic. But
because even a lot of universities don't even allow users to collect their own data. They have techs, right? So they're sitting, they're backseat driving, right? They've got an Uber for their they've got a self-driving, what do they call those damn stupid cars around San Francisco? They're not really driving themselves. Yeah, that's a really interesting point. Well,
Hmm.
we'll add that to the list of
Hmm.
of issues that people need to tackle. We're not the first field to tackle this kind of this issue, I'm sure. All right. Well, any other thoughts before I hit the old pause button?
That's pretty good. It's good.
All right.
Good to
Well,
catch up.
let
It's
me.
been a while.
Yeah, no. Well, let me pause. Let me stop this.